All right, let's dive right into this explainer. We're going to explore the absolutely fascinating blueprint of existence, taking a macro to micro journey through living beings and their structures. We'll be breaking down everything from the macroscopic kingdoms of life all the way down to the invisible microscopic structures that actually define it. It's like a giant biological puzzle, starting with the very building blocks of life itself. So, to kick things off, I want you to consider this massive paradox. Are viruses actually alive? It's a question that has honestly puzzled scientists for decades. Viruses exist on this incredibly thin borderline between living and non-living beings, a concept that, I promise you, will completely reshape how you view life by the end of this explainer. Keep this question in the back of your mind as we scale up and down the ladder of biological complexity. Looking back historically, early scientists, like Linnaeus, grouped literally everything into just two buckets, plants and animals. But, as you can probably guess, this simple system had some massive flaws. It completely failed to separate simple, nucleus-free organisms from complex ones, and it even threw fungi in with green plants. This biological confusion eventually led to Robert Whittaker's five-kingdom system, which was a real game-changer that completely redrew the tree of life. Now, what's really interesting about Whittaker's approach is that he didn't just group things randomly. He separated organisms sequentially from least to most developed, based entirely on their cellular complexity and how they get their food. We start at the bottom with Monera, the simple prokaryotes. Then we step up to Protista, move to nutrient-absorbing fungi, the photosynthesizing plantae, and finally Animalia, multicellular heterotrophs that actually have to consume others to survive. Zooming all the way in on that cellular complexity, we hit the prokaryote. This is essentially the simplest, most ancient form of life on Earth. A prokaryotic cell completely lacks a distinct, true nucleus and has absolutely zero membrane-bound organelles. Bacteria belong exclusively to this primitive, yet incredibly resilient category. In stark contrast, eukaryotes are a whole different ballgame. These are highly complex cells that boast a true nucleus, acting like a highly secure vault for genetic material, alongside specialized membrane-bound organelles. This structural leap is massive. It's the foundational cellular architecture that builds everything from your pet dog to the mushrooms in your salad. Now, peeking inside these complex cells, it gets even more fascinating. We run into cell inclusions. So what are these exactly? Well, they're simply non-living substances just kind of hanging out in the cytoplasm. Think of them as temporary or permanent storage units, like lipid drops or vacuoles. They aren't surrounded by membranes, and they don't directly engage in the cell's metabolic activities at all. But unlike those passive storage units we just talked about, cell organelles are the actual living, breathing machinery of the cell. Protected by their own specific membranes, they do the heavy lifting required to keep the cell alive. From the nucleus acting as the brain to the Golgi body acting as a shipping center, these are literally the vital organs of the microscopic world. Plunging even deeper into the microworld, we find microorganisms. These are the tiny, invisible beings that basically dictate the rules of life on Earth, despite only being visible under a microscope. Spanning across the Monera, Protista, and fungi kingdoms, these microscopic entities are everywhere, profoundly impacting both our environment and our own bodies. As you can see, the sheer diversity down in this microworld is just staggering. You've got your simple prokaryotic bacteria, and then you have the amoeba, a single-celled eukaryote that physically hunts its prey using false feet called pseudopodia. You have fungi, which are non-green organisms with a thallus body, like yeast or mucor. And finally, you have the highly unique viruses, which act as microscopic infectious agents requiring a host just to reproduce. Take a look at the bacteriophage, for example. This thing is biological machinery at its absolutely most ruthless. It features this crazy hexagonal capsid head protecting its DNA, sitting on top of a tail sheath. Those tail fibers recognize specific bacterial receptors, the sheath contracts, and it pierces the cell wall, injecting its DNA just like a microscopic syringe. It's wild. And we really need to understand this, because these microorganisms don't just stay neatly in textbooks, they actively invade our spaces. Bacteria can spread rapidly through airborne droplets or via contaminated water and food. Viruses utilize similar paths, but can also be highly bloodborne. Meanwhile, fungi often rely on floating airborne spores or direct skin contact. They are absolute masters of transmission. The destructive power of these organisms is, quite frankly, terrifying. Bacteria are responsible for historical heavy hitters like the plague, cholera, and tuberculosis. Viruses cause a vast spectrum of illness, from rabies and HIV to COVID-19. Amoebas can trigger severe amoebic dysentery. And fungi? Well, they're the culprits behind incredibly stubborn conditions like athlete's foot, ringworm, and aspergillosis. Okay, but it's not all doom and gloom. Believe it or not, microorganisms are actually the invisible backbone of human survival. In agriculture, rhizobium bacteria are out there fixing vital atmospheric nitrogen into our soil for leguminous plants. In industry, we rely on lactobacillus to ferment our dairy and pickles. And most incredibly, certain fungi, like penicillium, produce the very antibiotics we use to fight off the harmful microbes. Let's bring this microscopic science right into your own kitchen for a second. We just mentioned that pickles are created by good bacteria, right? But why exactly do your delicious jarred pickles sometimes succumb to a fuzzy, gross fungal attack? It turns out your pantry is actually a microscopic battleground. Well, fungi absolutely love warm, moist environments with access to oxygen. So, to win this microscopic turf war, you have to aggressively control the environment. You need dry, airtight containers and protective chemical barriers, like salt and vinegar. Storing them in the cold, using sterilized spoons, and adding a thin layer of oil on top physically blocks those oxygen-loving fungi from ever taking root. And this whole pickle defense strategy is really just one piece of the broader science of food preservation, where we basically manipulate the physical world to stop biological decay. Dry preservation uses moderate heat, like 20 to 30 degrees Celsius, to strip out the internal moisture that microbes need to survive. Wet preservation uses high concentrations of salt or acid as a barrier, and cold preservation basically freezes these germs into complete passivity. But when our preservation methods fail, the consequences are pretty severe. Microorganisms like Salmonella and E. coli can breed rapidly in our food, basically turning our meals into biological weapons. This triggers what we all dread: food poisoning, which is a violent bodily reaction to microbial contamination and toxins. This breakdown highlights the terrifying cascade of effects. Just simple and proper handling can lead to intense nausea and vomiting, while undercooked meats can introduce pathogens causing massive dehydration. But the absolute worst offenders are microbial and fungal toxins, like aflatoxins. These can cause everything from blurred vision to severe infections, leading to potentially fatal kidney failure, known as HUS. Now, the real irony here is that completely neutralizing these incredibly complex, dangerous pathogens often comes down to the most basic everyday actions: washing your hands, keeping raw meat separated from your veggies, strictly respecting expiry dates, and drinking properly treated water. It is literally our macroscopic behavior controlling these microscopic threats. Stepping back from our kitchens for a minute, when you look at the massive, wild diversity of these pathogens, fungi, plants, and animals, you quickly realize the absolute necessity for a strict system to categorize them all. Enter taxonomy. This is the meticulous branch of science dedicated entirely to the classification, identification, and naming of every living organism on Earth. To make sense of all this biological chaos, science uses the binomial system of nomenclature. Basically, every single identified organism gets a two-part scientific name. The first word represents the broader genus, and the second is the highly specific species. For example, humans are Homo sapiens. It's basically a universal map for the entire web of life. But here is exactly where taxonomy hits a massive, seemingly impenetrable wall. Remember the question we asked at the very beginning of this explainer: are viruses alive? Well, viruses are what we call obligatory parasites. They cannot carry out a single metabolic activity, no respiration, no growth, without hijacking a living host cell first. And here is the ultimate clash of definitions. Inside a host, viruses act completely alive, aggressively reproducing and passing on their genetic material. But outside a host, literally zero metabolism. In fact, they can be crystallized and stored in a jar just like ordinary salt. Because of this totally bizarre dual nature, viruses sit permanently on the borderline, completely defying the very definitions of biology. Which brings us to our final thought. If the very rules of taxonomy and biology completely break down at the microscopic level with viruses, what other bizarre rule-breaking structures are hiding just out of sight? What else is waiting under the microscope to completely redefine life as we know it? Keep questioning, keep exploring, and we'll catch you in the next explainer.